Okay, genetics notes part 3. I missed a couple of notes, but my board got so messy I had to kind of quit and come back. So I was talking about how different chromosomal patterns in meiosis, different inheritance patterns, can cause different issues. I didn't mention part B under number 6. It says changes can be harmful or lethal. Some affect fertility, and half of all miscarriages are due to chromosome abnormalities. These changes have accumulated in chromosomes of all species over time. And one specific one that I wanted to talk about, which is really interesting, are your X and Y chromosomes. But I'm missing number 1 here. Telomere, that's the word I'm looking for. So telomere section found in the middle of chromosome number 2 of humans is suspected of fusing 2 chromosomes together, because chimpanzees have like a 2A and a 2B, and so they are separate, and telomeres are the portions at the very tips of your chromosomes. You lose telomeres every time your DNA replicates, and so as you get older, one of the signs of old age is shortened telomere length, and it can actually be one of the causes of different diseases of old age. But so they have found that some of these telomeres that are typically at the end of your chromosomes are actually in the middle of chromosome number 2 of humans. And so they think evolutionarily, somewhere along the lines, humans have joined chromosome number 2, which in chimpanzees, like I said, is 2A and 2B. Excuse me. They have joined those two together and ended up with just one chromosome. So another one that is different are the X and Y chromosomes. X and Y chromosomes were once homologous autosomes. Homologous autosomes meaning that they affect the normal life, not just sex features. And this would be like in reptile-like ancestors of mammals. But the SRY gene mutated, and it resulted in reduced crossing over. So when they were no longer allowed to cross over like in prophase 1 and share DNA, they were devolved independently of each other, so they had their own mutations accumulated separately, and so they ended up in the X and the Y chromosome respectively. Moving on to DNA cloning. So DNA cloning is very important in today's sciences. They use restriction enzymes to cut a sample of DNA into pieces. DNA ligase splices fragments into plasmids, which are circular pieces of DNA from bacteria. And then new recombinant DNA are inserted into host cells, just like bacteria. The division of host cells then produces a large population of genetically identical cells, and reverse transcriptase, genetically identical cells, oops, I need to go back and finish that note, which are clones, each containing the DNA fragment. Then reverse transcriptase is used to transcribe RNA into cDNA or clone DNA for further cloning. So this is my lovely picture of what I just explained, and for some reason, there we go, we got all flipped around. So this is my human cell. They take EcoRI1 and they will go in and splice it. They will take that DNA and get it ready and insert it into a plasmid, and this plasmid will then go into the bacteria. The bacteria will then be grown in a lab. That plasmid or that DNA would be harvested, and then we can use the DNA for other things. So that's kind of like you harvest the DNA plasmid from the bacteria for this use, and then you grow and grow and grow more. And for example, human growth hormone, so people that have issues with their pituitary gland, this is how they make new human growth hormone for them, by doing recombinant DNA. And then it will produce the human growth hormone protein that they need in their bodies. Genetic engineering, part 8, almost done. Scientists use a DNA library containing different fragments of DNA to represent an organism's entire genome. They use tools like PCR. PCR uses primers to rapidly increase the number of copies of a targeted section of DNA without needing to clone live cells. So instead of having to clone live cells, if you just want to replicate a bunch of DNA, let's say you find just a little bit at a crime cell or a crime scene and you need to make a whole bunch more, you can use DNA polymerase chain reaction, or PCR, to replicate that. And basically, you're just falsely causing this DNA piece to replicate itself over and over and over and over again. Where am I at? I've lost my place. I'm sorry, guys. PCR uses primers to rapidly increase the number of copies of a targeted section of DNA without needing to clone live cells. Electrophoresis is what we did in lab this week. This is what separates fragments of DNA cut by restriction enzymes by length into bands. So it runs them through the gel, and that's where you saw the bands. Then the sequencing reveals the order of nucleotides in DNA. So sequencing is another type of genetic engineering. They've done a human genome project, which has sequenced the entirety of the human genome. And the human genome is approximately 3 billion nucleotides long. Genomics, which is a study of genomes, provides insights into the function of the human genome, and it looks for similarities between different organisms for evidence of evolutionary relationships. So it was who looked at our DNA and chimpanzee DNA and be like, oh, yeah, those things are kind of similar because of the location of the telomeres and what happens on the chromosomes. And so they've actually reclassified a lot of organisms based on genomic evidence. DNA profiling can also identify a person by SNPs, which is another thing we talked about in lab today, single nucleotide polymorphisms, which is an array of short tandem repeats. So you can get a DNA fingerprint of a person, which is basically a number of bands that show up in DNA electrophoresis, and compare that against DNA from a relative or DNA that would be collected from a crime scene to identify a person. You can take one gene from one species and insert it into an individual of a different species, or you can modify that gene and insert it into the same species to create a GMO. So all of these different things, genomics, GMOs, DNA fingerprinting, electrophoresis, sequencing, PCR, all of these are different ways of looking at the genome through science technology and identifying it in different ways. Bacteria and yeast are used to make proteins with medical value, like I talked about when we talked about the DNA cloning and making the human growth hormone. Transgenic crops such as corn and soy provide pesticide resistance and increased nutritional value. Now those have pros and cons. Increased nutritional value is awesome, but pesticide resistance means that you can spray these crops with all kinds of pesticides to grow them in mass. But are they really safe to eat? So there's some debate on whether having extra pesticides in your food is a good thing and how much of that actually gets onto you. I know if you go through these GMO crop fields, not because of the GMOs themselves, but because the amount of pesticides sprayed on them, you have to wear a full-on hazmat suit with a respirator so you won't get very sick and die. Finally, gene therapy is a way of transferring a gene into body cells, like human body cells, to correct a genetic defect or treat a disease. But if you're looking at gene therapy, so you're modifying your actual genome, it would be like if you, or for my husband, for example, with polycystic kidney disease, if he could get a way of inserting the correct gene into his body, the recessive one, that would not cause his kidneys to make any more cysts, and we could change the ability of his kidneys to be normal and function normally without making cysts. But we do that by changing his genome. So give his kidneys like a virus that causes it to spread and change it. Is that okay or not? And the reason being why you would even wonder whether that's okay is because while you do have potential benefits, you have to weigh against risks like eugenics. So are we going to have the ability to create a master human race, and who gets to decide what that race looks like? Who gets to decide who lives and dies, and what genes are worthwhile of keeping and what need modified? So those are your notes for chapter 13, 14, and 15 about genetics, chromosomes, and genetic engineering.